My name is Eric Chow, and I'm from the University of California, San Francisco. And today I'm going to give you a talk about single-cell sequencing. So, brief outline, we'll cover what single-cell sequencing is with a big focus on RNA sequencing. We'll cover plate-based methods, microfluidic methods, combinatorial indexing methods, and then talk about other single-cell sequencing methods that don't necessarily relate to RNA. So, a big question that comes up is, why do single-cell sequencing? And a good analogy of this is this fruit smoothie analogy. So let's say that you're working with a blood sample and you want to take a look at the expression profile within that blood sample. So blood is a complex mixture of many different cell types. You have B cells, T cells, macrophages, and neutrophils, similar to a smoothie where you have lots of different types of fruit. And let's say you're really interested in the characteristics of, say, a raspberry or an orange. But instead of analyzing that raspberry or orange, you mix it up with a pineapple, a banana, some strawberries, blend them all together, and then that's what you're tasting. It's going to be very difficult to figure out what tastes are coming from that raspberry. And so the same thing when you're trying to analyze data from a single cell versus a bulk sample. It's really advantageous if you can get data specifically from individual cells as opposed to making a bulk measurement. And one really cool thing about single-cell sequencing is that with the data you can identify lots of different cell types. And so this is a representation of a data set from a blood sample. And in this case, many thousands of single cells were processed together in parallel. Each of those individual single cells transcripts were sequenced and analyzed. And you can detect up to the 20,000 different genes that are present in the human genome. And so it's really hard to depict data in 20,000 different dimensions. So one common method is to collapse that down into 2 dimensions using dimensionality reduction. There are many different methods, which we won't be able to go over today, but you end up with a plot that looks like this, where each small dot represents a single cell, and these dots are clustered, and cells that are clustered closer together are more similar to each other. And so you wind up with these clusters of different cell groupings that are represented on this plot. And so this by itself isn't useful. You can tell that you have different cell types there, but you have no idea what they are. But you can dig into the data. And for instance, if you can go into each of these clusters and ask, what are the transcripts that differentiate, say, this cluster at the top from this cluster next to it, and you do this for the different genes, you can use this information to then classify the different clusters and identify them. So for instance, in blue, you have B cells. In red, you have activated CD8 T cells. You have dendritic cells in this purple color, CD4 monocytes in green, NK cells in gray. And the list goes on and on. You can identify lots of different cell types by looking at the transcriptional profiles within each of those clusters. And so if you've watched some of these flow cytometry videos on iBiology, you'll note that you can get the same information from flow cytometry. So why do single-cell sequencing? And one reason why is that these clusters can be further broken down and analyzed by those genes. And so again, we can make measurements on up to 20,000 differentially expressed genes that might be present in samples. Whereas with flow cytometry and mass cytometry, we might be limited to a few dozen markers, and that's it. And so for instance, if we were to break down this cluster and ignore the rest of the clusters and just analyze this cluster and try to differentiate this a little bit more and zoom in and recluster the data, we can take that green cluster, recluster it to get some better separation, and then ask what are the genes that separate these. And you can go through this process iteratively. So if you identify an interesting cluster, you can dig down to see whether a gene expression signature is specific for a subset within a cluster. And this is really the power of single-cell sequencing, is that you get a very high dimensional readout of the data that you can't get with other methods. And so the output of single-cell sequencing methods has really increased significantly over the past decade. So initially, back in 2009, was the first study where they sequenced one cell, and that was it. And over the years, you can see that there's been an exponential increase in the number of cells that have been processed in different studies, with each of these circles representing a different publication and the number of cells analyzed. And so we'll start looking at the techniques that were used about 5 years ago and earlier that really analyzed individual cells by sorting them into separate wells of a plate, preparing libraries from each of those individual cells, and then sequencing them. The most common method was this plate-based SmartSeq technique. And in this technique, you use a flow cytometer and you sort your cells of interest, one cell into each well of a 96 or 384 well plate. You lyse that cell, and then you do a reverse transcription reaction using a SmartSeq style library preparation. And with this library preparation method, you have a oligo DT that'll prime a reverse transcription reaction off of mRNAs that are polyadenylated. This oligo DT has a handle on the end, and during this reverse transcription process, DNA gets made using RNA as a copy. And at the end, with certain reverse transcriptases, they'll add a couple of non-templated C bases. And these C bases are used in conjunction with a template switch oligo that has these Gs at the end. So this template switch oligo on top hybridizes on to these non-templated Cs got added, and the reverse transcriptase is able to use this template switch oligo as a template for further polymerization. And in the end, you wind up with a cDNA that has PCR handles on both ends. These undergo amplification so that you get many, many copies of each cDNA. And then this goes into a sample preparation, a DNA library preparation method to complete the library prep process. And that's depicted here. And the most common one used is this Nextera DNA library preparation method, where you would have your amplified cDNA up here. The Nextera transposase will fragment that cDNA into smaller bits, add partial Illumina adapters, and then through PCR, you amplify up this material and complete the library preparation process. And so with those methods, you were able to process a few hundred, maybe a few thousand samples with a lot of work, because you were processing each cell in a separate tube of a well of a plate. And so this required a lot of pipetting, a lot of manual work, and is fairly expensive because you're essentially doing a separate sample DNA library sample preparation for each cell. And so a few years ago, there were a few methods that came out that parallelized this process, taking advantage of different techniques that we'll go into next. And the first one we'll cover is the method Dropseek. So this is a method that uses microfluidics, or essentially different methods of creating very, very small volume droplets and doing molecular biology within those droplets. And in this method, you have a chip where you have beads that flow in from the left-hand side. Cells will flow in from this first junction, and so they'll get mixed together. In the next junction, you have oil that flows in, and this oil will cause this aqueous phase to pinch off and form an emulsion. And because of the way the system works, you wind up with a lot of droplets at the end. Most of those droplets will be empty. Some droplets will contain a bead, some droplets will contain a cell, and a small fraction will contain both a bead and a cell. And that's depicted over here. And again, this is a small minority of the droplets will contain both one cell and one bead. What's special about these beads is that each of these beads has many bar-coded oligo DT oligos on the surface. This will allow it to capture the RNA that comes from each of the cells that are co-encapsulated. And again, each of these beads has a different barcode from all of the other beads. And so what happens is you get capture of the RNA onto the beads. After that, you pull down the beads out of the solution and you do a reverse transcription reaction, again with this template switching process. And at this point, all the contents of each cell are stuck to one bead, and all of those cDNAs have the same barcode because they're attached with the same bead. And again, that bead has many oligo-DTs with that same barcode on there. You can amplify up the cDNA just like we discussed before by using PCR. And then you can prepare a DNA sequencing library that'll sequence the ends of all of these transcripts. And because we've tagged each of these transcripts with a barcode that corresponds to a cell, well, essentially we've been able to process many, many cells in parallel in a couple of tubes. And with this method, you can sequence thousands, even tens of thousands of samples or cells in a single sample using just a couple of tubes. So you wouldn't need 30,000 different wells, which would be incredibly laborious and logistically just almost impossible to do. So this was a commercial effort that came out of that came out of an academic lab. And one issue with this method is that, again, most of those droplets contain nothing, and only a small fraction of those droplets contain both one cell and one bead. And the reason for this is because those cells and beads randomly get encapsulated into those droplets. And so it looks something like this, where you may have a lot of partitions that are empty in gray, and then you have a fraction that have a single bead or a single cell And then because of the random nature, although you don't have most of the wells or the droplets filled with a cell or a bead, you do have some droplets that contain 2 or more beads or 2 or more cells. And so you could do something where you increase the concentration of cells and beads so that you get more single droplets or droplets that contain a single cell or a single bead, but your doublet rate increases by a lot. And there's no way to really distinguish this from the sequencing data. So this is the reason why with this Dropseek technique, both the cells and beads are loaded at a low concentration to avoid those multiplets. So about a year or 2 after Dropseek came out, 10x Genomics, a commercial company, came up with a very, very similar method. If you take a look at it, it looks extremely similar. You have barcoded beads that flow in from the side, cells and reagents come in, and then those get mixed, and then oil comes in to generate an emulsion. But you can see in this picture, most of those droplets contain a bead, and it's because they were able to engineer both their microfluidic device and these beads so that greater than 90% of the droplets contain 1 bead and only 1 bead. And so this increased the efficiency by a lot because they don't have to load the beads at a low concentration. Another difference with this technique is that inside of these droplets, you don't just have capture, but you also undergo reverse transcription within these droplets, which is one other difference. So at the end of this reverse transcription reaction, you break the emulsion, you take the cDNA and amplify up in a single tube, and then you construct your sequencing library again in a single tube, and then you can sequence this on the sequencer. And depending on how many cells you load in the platform, you can sequence up to 25,000 cells in each of the lanes of this chip, and this chip has 8 lanes on there. So potentially in a single chip, you could process 200,000 cells. And this process on the chip actually only takes 15 minutes. The downstream portions over here take about half of a day, but that's pretty impressive that essentially in one long day, you could process 200,000 cells at a time, or more if you ran multiple chips. So another method to parallelize this process of generating single-cell sequencing libraries is to use microwells. So instead of using an emulsion to separate your cells from each other, you have a really, really tiny array of wells. So each of these wells may only be a few tens to a few hundred microns in diameter. And so there are a couple of open source and also commercial platforms that do this, such as SeqWell, CellC, and ones from BD. And essentially the way these work is that you have this microwell array, and there can be thousands, tens of thousands, or even hundreds of thousands of wells on a chip. You lay down barcoded DNA beads. In this case, these are colored blue, and these beads are engineered so that most of the times you get 1 and only 1 bead into a well. They kind of coat this array of microwells, and then you apply your cells on top. In this case, in this example from CellC, these cells are labeled in red so you can see them. And again, in most of these wells, you have a blue colored bead that's present, and then the cells will just settle down into the wells. And then you go through lysis and all the enzymatic reactions to tag the RNA from each of those cells with a unique barcode, go through this library preparation process to generate cells, and so to generate sample libraries. And with some of these methods, they have wells, again, that have hundreds of thousands of wells and potentially 1 million or more wells on a chip so that you process many, many samples. So within this past year, a paper came out that had over 1 million cells processed in the study. And this method actually used a method called Combinatorial Indexing, which I'll go over next. And so with Combinatorial Indexing, what happens is you do a lot of in situ reactions, which means you use a cell as a compartment. And you're not working with individual cells at any given moment, but you do multiple rounds of barcoding and mixing so that you generate this combinatorial barcodes that will allow you to generate single-cell data. And this is a method from an academic lab at the University of Washington that used the combinatorial indexing method. And the way this works is that you have fixed cells on the right. So these are fixed and permeabilized cells so that reagents can flow in and out of the cells. You sort maybe a few dozen cells into each well of this 96 well plate. So you're not dealing with single cells. There's still multiple cells in each well of the plate. You undergo a reverse transcription reaction with a barcoded oligo DT. So in each of these wells, you have a different barcoded oligo DT. So all the cells within each well will have the same barcode at this step. okay? And this is depicted by these colors over here. So let's say you pulled from 3 wells, and so you have many cells that have green, blue, or red. You combine these cells together, you shuffle them up, and you randomly redistribute them into a new set of wells of a 96 well plate. At this step, you take your cDNA that's present in each of those cells, and then you do a different reaction that will then add a second barcode. Again, each of the cells in each well will receive the same barcode, but chances are each cell in this well came from a different well in this first plate. And so with this combination of barcodes, with 96 and 96, you have about 10,000 combinations. If you're only sequencing about 1,000 cells, you don't have a very high chance of having 2 cells with the same barcode. And this is essentially what the final library looks like. You have a RT barcode, or reverse transcription barcode. You also have 2 different barcodes that are in the Illumina adapters that can be used as well. So you can potentially barcode up to 3 times with this method. And the power of this technique really comes in as you add more of these levels. So for instance, again, if you had 2 sets of 96 well plates and you're barcoding them, you have almost 10,000 combinations. If you were to work with 384 well plates, you'd have almost 150,000 combinations. And it actually isn't ridiculous to work with several 384 well plates at each step. And so if you had 4 384 well plates at each step, or 1536 combinations at each step, you can generate about 2.4 million different combinations. And so this is just with 2 rounds of barcoding. If you were to do 3 rounds of barcoding and add another barcode, in this case 96 or 384 for a 3rd level, you really scale the numbers. And so in this case, I'm actually representing these numbers in millions now, just because the numbers would be too big to fit on this chart. And so in this case, if you were to do 96 by 96 by 96 with 3 levels, you'd have about 1 million combinations. If you did 384 by 384 by 384, you'd have almost 60 million combinations. And so this is the reason why this scales really, really well, especially if you add that 3rd level of barcoding. So changing gears a little bit, we focus mostly on RNA sequencing, but it is possible to do other things with single-cell sequencing platforms. And one of those is to quantify proteins. And so you might ask, how do I quantify a protein with DNA sequencing? And the method that's been used called CytSeq is that you have your antibodies that will bind onto proteins of interest. So this is similar to an antibody that you might use for flow analysis, but instead of conjugating a fluorophore to be read out by flow or an isotope for CyTOF, you conjugate on a DNA barcode. And so this DNA barcode has a PCR handle at the end, a barcode that tells you which antibody is attached to you. Again, this is just a DNA sequence barcode. So your A, C, Gs, and Ts, you just have some unique sequence for each antibody. And then a poly A tail. So this poly A tail will allow this antibody sequence to get captured by the single-cell sequencing methods. And then you can read out the barcode essentially as a transcript that'll tell you how many copies of this antibody were bound to that particular cell. And so this is just some data comparing RNA analysis with protein analysis, just using human and mouse cells that have been kind of mixed together to show you that the method works. And there are currently panels of antibodies, about 100 or even more antibodies that you can use in conjunction with single-cell sequencing. And the cool thing here is that you can get not just protein abundances for those proteins that you have antibodies for, but you also get the transcriptional readout in terms of which genes are also turned on and off in each of these cells. So you get this kind of a multi-omic approach where you can analyze both protein and RNA. So one issue I told you about earlier with some of these methods is this issue of multiplets. So if we ignore just bead multiplets and just focus on cell multiplets, this is one reason why we're limited in terms of the number of cells that come out of these both these microfluidic and microwell platforms. And again, you don't want to load very many cells because you want most of these droplets to be empty so that you avoid these doublets. So again, if you were to increase the concentration of cells that you load in to maximize the number of singlets, so for instance, over here you have 4 singlets, you do increase the rate of your doublets so that these increase here and here. Now, if there's some way to identify these doublets, you could remove them computationally from your data analysis. And so, for instance, if you had 2 differentially labeled samples that you pull together and then you flowed into your microfluidic device at a high concentration, you would get multiplets. But if the multiplets came from 2 different cell types and you can identify that, you can exclude those from analysis and then actually get more single cells for the same cost. And so one of the first strategies to do this was a method called demuxlet. And this was multiplexing by genotype. So this group here at UCSF was working with different patient samples. And with different humans, we all have different DNA sequences. And some of these sequences are found in our transcripts. And those can be detected by single-cell RNA sequencing. And so this example, they had 8 different donors that had different genotypes by different colors here that got combined and then run in a single-cell sequencing platform. So you get a whole bunch of single-cell data. And if you take a look at each individual cell, let's take a look at one cell here, we see a certain genotype in the transcripts, and we can match that with one of the original donors. So we can tell that this sample or this cell came from this patient sample. And you can do this for most of the cells in the data set. What's really cool about this method is that if you have a doublet, 2 cells in a single droplet, and you have 8 different samples, chances are those 2 cells came from different samples and they'll have 2 different genotypes. So if you analyze the data within those cells, if you see the presence of 2 different genotypes, you know that's a doublet, and we can remove those from analysis. So this allows you to bump up that concentration and get more single cell data for the same cost. So if you're not working with genetically distinct individuals, this method won't work. And so there are a couple of non-genetic strategies that came out. The first one was from the same group that developed CytoSeq. They realized that if they use an antibody that's binds to some type of universal surface antigen on cells, they could take that antibody and split it up in a couple of different aliquots, and in which each aliquot, you can give it a different barcode. Then you can take your different samples and then add them to these different aliquots of antibodies to allow them to bind. At that point, you can pull them all together, run them in the single-cell sequencing platform, and you look for the presence of which barcoded antibody showed up. And this will allow you to assign each of these individual cells to a particular starting sample because you knew which barcoded antibody used for each sample. And again, this also has the advantage where if you have 2 or more cells in a droplet, you can detect those because you'll see 2 different barcode sequences assigned to a single droplet. And you can exclude that from analysis. More recently, in collaboration with another group at UCSF, we developed a method that is a universal method that uses lipidated DNA that embeds into the membranes of cells. So this doesn't depend on a surface antigen. It just depends on an accessible membrane that this barcoding DNA molecule can insert into. So it inserts into the membrane of cells. You can insert a different barcode into each pool of samples and then pull them together. In this case, we were able to barcode 96 different samples, pull them together, and we can identify all the singlets and the doublets. And you can see all different 96 samples represented here in this plot. So in conclusion, there are actually many different types of single-cell sequencing. I've only focused on RNA today, but there's DNA, there's protein, there's chromatin accessibility, and many, many new methods coming out every few weeks. So it's an exciting field to be in. Single-cell sequencing also generates high-dimensional data, especially if you're looking at RNA, because you're probing the quantity or the expression level of all 20,000 different genes that can be expressed in cells. And so this can reveal biology that's hidden from lower dimensional techniques. So single-cell sequencing does have some disadvantages. It is lower throughput and more expensive compared to flow cytometry and mass cytometry. With those methods, you can easily analyze hundreds of thousands or millions of cells at a time. And I also didn't mention single-cell analysis that doesn't use sequencing. So there are microscopy-based techniques that are out there as well that have some very interesting properties, including the ability to give you some spatial information. So if you're working with tissues, with most single-cell sequencing techniques right now, you have to dissociate those tissues into a single-cell suspension and then run them. So you lose a lot of that spatial context that might be important for the biological question that you're trying to answer. And some of these microscopy methods can offer you some additional information that might be valuable. And so again, thank you for your watching today, and hope you enjoyed this video.